Hello everyone, my name is Priyanka. My qualification is MSc B.Ed and CSIR NET. Today I am going to explain interaction of genes. What is interaction of genes? But first, before going to interaction of genes, we have already understand interaction of alleles in the Mendel's law of inheritance. What happened there? In the first law of Mendel, that is law of segregation of gametes, what happened at the time of gamete formation, one allele go into a separate gamete. If one gene has two alleles, then one allele will go into one gamete. For example, in human being, we know that human had 22 pair of autosomes and one pair of sex chromosomes. For example, any human male have 22 pair of autosomes plus X and Y sex chromosomes. What happens when there is going to be formation of gametes? Well, all the sperm will have one pair of autosomes and one X, and other sperm will have one pair of autosome and Y. Not both X and Y will go into one sperm. Only either X or Y will be present with the autosomes. That is called purity of gametes. It means at the time of meiosis, half the chromosomes will go into one gamete. Second law was law of independent assortment, where we took two characters in consideration at one time and two alleles of one gene. For example, seed shape. For seed shape, we have wrinkled and round, and for seed color we had yellow and green, and both four combinations will appear in children or progeny of the parents, but some new combination will also be present. How this new combination is going to form in the progeny? Because at the time of gamete formation, there is independent assortment of alleles. So the interaction of alleles in the Mendel's law is at the gene level, but here we are not going to understand interaction of alleles. We are going to understand interaction of genes. When two or more genes will control one character, and by the interaction of those genes we will have many characters for one thing, then it is called as interaction of genes. For example, in Mendel's law of independent assortment, we consider two genes controlling two characters. But it's not necessary that a single character is controlled by one gene only. Instead, it may be controlled by more than one gene. In such a situation, two or more than two genes may interact to give rise a particular phenotype. Here, we are talking about interaction of two or more than two genes which are responsible for one phenotype or one character. In Mendel's law, we were talking about different genes and different characters at one time. There, one gene was controlling one character and one gene was having two alleles. But here we are talking about two to more than two genes at one time affecting only one character. That is difference. For instance, if gene A is responsible for phenotype yellow and gene B is responsible for phenotype gray, both A and B when present together may give rise to a different phenotype. That is not gray, not yellow, it may be different. Both gene A and gene B will still obey the principle of segregation and independent assortment. In the Mendel's law, alleles were following segregation and independent assortment, but here genes will also follow segregation, law of segregation and law of independent assortment. If A and B are two genes, both dominant over their respective alleles A and B, then interaction will depend upon. Here we are taking an example. In this example, A and B are two genes and they are controlling one particular character in one organism. Then 4 types of interaction or 4 type of conditions these both genes will give, will cause. What are 4 conditions? In first condition, the presence of both dominant alleles. It may happen that when both the dominant alleles are present in one individual, then it will give a distinct phenotype. In second condition, the one dominant allele, for example, A may be absent but B may be present. In third type, the absence of B may be absent or A may be present. And in fourth condition, both A and B are absent. And it means only recessive alleles are present here. Recessive alleles means the recessive of A and recessive of B. Interaction is possible between more than 2 genes also, but for simplicity, interaction involving 2 genes will be considered here. Here, for example, when A and B are 2 genes, then the genotype, the AABB homozygous, A capital A small bb, that is this one, AA, one B capital, one B small, and AABB. These 4 type of genotypes will give rise AB phenotype. It means that whatever phenotype, when one capital A and one capital B will give rise to this organism, these 4 will also be same, because in these 4 genotypes, one A will always and one B will always be present. Here we can say one A is also present, one B, 2B is present, and but the phenotype will be like this, AB. For these 2 examples, the phenotype will be of this type, when one A dominant gene is present but one dominant is absent, which we saw in my previous slide. There were 4 conditions. When both dominant are present, then the phenotype will be this type. When one dominant will be present and one recessive will be present, then phenotype will be this type. In the next category, first dominant is absent but second dominant gene is present, then phenotype will be this category. And in fourth condition, both recessive are present, not any dominant, then phenotype will be this category. And the ratio is same as dihybrid ratio of Mendel's law, Mendel's cross, 9:3:3:1. So two gene pairs affecting same character. In Mendel's dihybrid cross, dihybrid ratio was considered where the 2 genes were independently controlling separate characters. Therefore, there was independent assortment of the genes as well as characters. In case of comb shape in poultry, this is very good example of interaction of genes. In comb means when you have seen a hen, it has a crown shape, red color crown shape, crown on its head, that is called comb shape. In poultry, 2 genes are responsible for these characters. While one gene gives rise to rose comb, for example, one gene will give rise to rose comb, another gives rise to pea comb. Two genes are responsible for comb shape in poultry. Second will give rise to pea comb. Each of these two is dominant over single comb, and the third scenario is single comb. When single comb will be formed, when there is all the recessive genotype. When there is no dominant allele is present, then the phenotype will be single. However, when both of these brought together, a new phenotype walnut appears. It means that when the genotype is RR, only R type is present, then the comb shape will be rose. When PP will be present, comb shape will be green, P shape. When both the dominants are absent, only recessive are present, then the comb shape will be single. And when one R and one P are present, it means heterozygous, then it will have walnut. It means by interaction of two genes, we are having 4 phenotypes of one character. When both dominant are present, then the comb shape will be walnut. Only R is present, one gene type is present, the comb shape will be rose. Only P dominant P is present, comb shape will be P comb. And when both the dominant are absent, then comb shape will be single. So this is the example of interaction of genes in comb shape in poultry. Here we can see when both the recessive alleles are present, comb shape is single. This is called single. When one only capital R, one dominant gene is present, it is rose. Here one this is single genotype. Only recessive are present. Here this is P shape, P. Here one capital P is present. But R is recessive. R is not dominant here. In this rose, R is present in dominant form, but P is not present in dominant form. Here P is present in dominant form at least, but R is present in recessive allele. Here both the are recessive, that's why it's a single. And here walnut, when at least one R and one P are present together, then it is walnut phenotype. And here it's written here, when both R and P dominant are present, it will give walnut phenotype, and this will be 9:3:3:1. Single or recessive will be 1. Like Mendel's dihybrid cross, recessive was only 1. Here it is also only 1. As worked out by W. Watson and R.C. Punnett, when both dominant alleles are present, walnut phenotype appears, and when both recessive alleles are present, single comb appears. Rose and P each appears due to the presence of different but single dominant alleles. If P and rose are crossed, or if walnut and single are crossed, the F1 individuals in both the cases will have walnut phenotype. It is same like a dihybrid cross we have read in the Mendel's law of inheritance, where in the F1 is always heterozygous, like this, that is walnut. These individuals, when crossed among themselves, like self-fertilization, when we read Mendel's law, we know that F1 was self-fertilized, or fertilized by heterozygous condition. These individuals, when crossed among themselves, will give 4 phenotypes in 9:3:3:1 ratio. This ratio is different from Mendel's dihybrid ratio. Why? Because there it represents 4 combinations of 2 pairs of characters, while in this example they represent 4 phenotypes of same character. It's a very important point to remember. 9:3:3:1 ratio is not same as Mendel's dihybrid ratio. There in Mendel's cross we took 2 characters at one time, and for two characters, seed shape and seed color, we take 2 different traits like yellow and green and round and wrinkled. It means 4 traits were there, and they were giving new combinations in 9:3:3:1 ratio. But here it's different because it's not combination, it's new phenotypes which we are taking here. 2 pairs of character, while in this example they represent 4 phenotypes of same character. Here we are having in the Mendel's law 2 gene pairs which are responsible for 2 characters are giving rise to new combination. Here 2 genes are giving rise to new phenotypes of one single character, that is comb shape in poultry. It's different. Epistasis. Epistatic interaction means that one gene masks or changes the effect of the other gene. In the interaction of genes, the first example is first most prevalent type of interaction of genes is epistasis. What it means? Epistasis means when one gene is masking the effect of other gene, and while masking it is giving new phenotype. If it would not mask the effect of gene, then new phenotype will not appear. But here by masking, one gene is masking the other gene, the new phenotype is appearing. That is called epistasis. In other words, it doesn't allow the other gene to express itself fully. While dominance works at intra-allelic but intergenic level, when we read Mendel's law, we talked about dominant and recessive. And dominant and recessive in what terms? When we take length of plant, we write 2 types of alleles, T for long and T for dwarf plants. It means we were talking about one gene here we are talking about one gene which have 2 alleles. One allele is for height of the plant, which will give tall phenotype, and one allele will give dwarf phenotype. It is at interallelic level or intragenic level. Intra means inside one gene. But epistasis works on intergenic level. Epistasis works when one gene is affecting other gene, not its allele, other gene. There is a difference between gene and allele. One gene have 2 or more than 2 alleles, and various forms of one gene is called alleles. For example, there is a gene that is called alpha, then its various form means alpha dash, alpha double dash is called as alleles of alpha. It is same because it's controlling the same character. Alleles are controlling the same character but giving rise slightly difference in that character. For example, alleles of length, that is dominant T, is increasing the height of plant, and small t, that is called recessive allele, it's not increasing the height of plant. That's why plant become dwarf. But actually both the alleles are affecting the length of plant, so they will be called as alleles or various forms of one gene. you should be clear about what is allele and what is gene, then you can understand this topic well. Otherwise you will get confused. I have made a video on this topic. If you want to check, you can go and watch. See, epistasis works on intergenic level. It affects other gene, not its allele. Epistasis can be of following two types. A masks the effect of B when a recessive allele is masking the effect of other dominant gene. It is called as recessive epistasis, and other is dominant epistasis, where A is masking dominant allele is masking the effect of other dominant gene. For example, A masks the effect of B, capital A masks the effect of capital B. In first one, small a recessive allele is masking the effect of capital V, but in second, the dominant A is masking the effect of dominant B. In some cases, this may be in one direction only, meaning that either A masks the effect of B or vice versa. But in some cases, both the genes become mutually epistatic to each other. It means that mutually epistatic means one gene is masking other gene and the second gene is masking the effect of first gene. Both are masking each other's effect. That is called mutually epistatic. Contrary to the above definition of epistasis, in population and quantitative genetics, epistasis refers to all non-allelic interaction. It is very important, non-allelic. Epistasis is non-allelic. It means it doesn't work on allelic level. It works on gene levels. One gene is masking other gene. One gene is affecting other gene. And other example of epistasis are complementary genes and duplicate interactions. You will learn about this also. Example of epistasis. In mice, the wild body color is known as agouti. In mice, wild body color is known as agouti. Agouti. There is a body color called agouti, characterized by banding of individual hairs. This mice will have banding of individual hairs. This will have striped hair on its body. The agouti color is controlled by a gene, say A. This agouti color is controlled by A, and it is hypostatic to recessive allele C. Hypostatic means this small c or recessive C can mask the character of A. It means it is small c is dominant over A. So A will be called as hypostatic to C, and C will be called as epistatic to A in relation to each other. Who is masking, that will be called as epistatic, and and whose effect is going to mask by other gene, it will be called as hypostatic. Hypo means inferior. The dominant allele C in the absence of A gives colored mice. When the dominant C is present, it will give colored mice. Colored. Moreover, in the presence of dominant allele C, A gives rise to agouti. Agouti color will develop when C will also be present, but not small c, dominant C. If one small c is present, it's okay, but one dominant is required for the agouti pattern. It means when both dominant A and dominant C will be present, then mouse will have agouti characteristic. And when only C will be present, then mouse will have colored hairs. Therefore, CCAA, this capital C, small a, small a will be colored, and capital C, small c and capital A will be albino, where both the recessive C and one capital A will be present. It is called as albino. Albino means no color. And why? Because this is masking the effect of A. This small c, recessive C, is masking the effect of A. Let's see in the diagram. Here you can see that when both capital C and capital A are present, then the body color will be a goatee, means stripe will be present on the mice. When only capital C is present but small a is present, then the mouse body color will be colored. When one small c and one capital A is present, here we will see the epistasis, because this small c is affecting the epistatic, doing epistasis to the A. Small c is affecting the effect of A gene, and that's why this C, small c and capital A is giving albino or not giving any color. In this example, this A is giving striped cat, helping in striped pattern, but here this A is not giving any color because of this small c is masking or hiding the effect of this capital A. That's why this CA and this small c and capital C both are giving albino, and then we will get 9:, ratio of 9:3:4. Why? If this small c would not have affect the capital A, then this ratio is not appeared. We cannot get this ratio 9:3:4 because of masking the effect of capital A. Because small c is masking the effect of capital A, we are getting albino here. And then 9:3:3:1 ratio is converted into 9:3:4, because this 3 and this 3 and this 1 is giving this 4 albino. This is example of epistasis. I hope you have understand well. This was today's slide. So thank you for listening me. We will meet in my new topic.